Welcome to today's webinar. My name is Chloe, and on behalf of Oxford Global, I would like to thank you all for joining us today. Today's webinar, Advancing Immunotherapy, the Critical Role of GMP-grade cytokines in Cell Therapy, is brought to you by Ritwika Biswas, Field Application Scientist at Sino Biological. After the presentation, we will lead into a Q&A. Please submit your questions through the chat feature. The full recording of the webinar will be made available to download on our secured website, and the details will be sent to you shortly after the webinar. Let's begin. Ritwika, over to you.
Thank you very much for the introduction. Welcome everyone, and thank you for attending today's presentation. My name is Ritwika Biswas, and I'm a field application scientist at Sino Biological. Today, I'll be discussing the critical role of GMP-grade cytokines in advancing immunotherapy with a focus on their application in cell therapy. Let's start by briefly outlining what we will cover today. In this presentation, we'll begin by exploring adoptive cell immunotherapy and the product development process in cell therapy, focusing on how these therapies are designed, modified, and scaled for clinical application. Then we'll delve into the importance of regulatory guidelines for raw material safety and discuss the challenges in cell therapies and supplier evaluation. Also, key consideration that ensures safety, quality, and reliability in advancing cell therapies. So, for some context, let's discuss some background for adoptive cell immunotherapy. So, the immune system is composed of two main branches, the innate and adaptive immune responses. The innate immune response is often described as the body's first responder, as it's fast, nonspecific, and it's what kicks into action when the body detects an invader. This includes physical barriers like skin and mucous membranes, as well as cells like macrophages, dendritic cells, and natural killer cells. However, the innate system isn't always enough to eliminate threats like cancer, which is why we need the adaptive immune response. The adaptive system is slower to activate, but highly specific and powerful. It has memory, and that means that it can recognize and respond more effectively to pathogens or cancer cells if they reappear. This long-term immunity is what vaccines, for instance, are built on. In the context of cancer, the adaptive immune response relies on antigen-presenting cells, such as dendritic cells, to activate T cells and B cells. These adaptive cells are responsible for recognizing and destroying cancer cells based on highly specific markers. Understanding this system is critical for developing immunotherapies that can harness the power of both innate and adaptive immunity to fight diseases like cancer. Cancer cells can trigger both innate and adaptive immune responses. So when you examine cancer, you see both the innate and adaptive immune systems are challenged by the complexity of tumor biology. Tumors are not just collections of rapidly dividing cells. They are actively invading the immune system. On the innate side, cancer cells express stress-induced proteins like MIG-A and MIG-B, which alert NK cells, macrophages, and certain cytotoxic T lymphocytes to mount an immune response. However, tumors often evolve mechanisms to evade or suppress these innate responses, making it difficult for the body to naturally control tumor growth. On the adaptive side, dendritic cells play a pivotal role in priming the immune system to recognize and attack cancer cells. This is where cytokines such as IL-12 and IL-18 come in. They act as messengers to help activate T cells, particularly CD4 helper T cells and cytotoxic CD8 T cells. The challenge lies in the fact that tumors can suppress or hide from the adaptive immune system, often by altering the presentation of antigens or by creating an immunosuppressive environment. This is why boosting the immune response through therapies like checkpoint inhibitors, adoptive T cell therapies, or CAR T therapies have become such a vital area of research. And we aim to overcome these mechanisms that cancer uses to evade immune destruction, allowing the immune system to target and eliminate tumors more effectively. Now, immunotherapy leverages the immune system to combat cancer, and it represents a paradigm shift in the cancer treatment area, because unlike traditional approaches like chemotherapy or radiation, which target the tumor directly, immunotherapy works by empowering the body's own immune system to fight cancer. One of the most exciting aspects of immunotherapy is that it offers a personalized approach, meaning it can be tailored to each patient's immune profile. It's basically like teaching the immune system how to recognize and attack cancer cells more effectively. So there are several types of immunotherapy. Each is designed to enhance or modify specific aspects of the immune system. For example, checkpoint inhibitors work by blocking the proteins that prevent immune cells from attacking cancer cells, essentially taking the brakes off of the immune response. And cancer vaccines, another form of immunotherapy, are designed to prime the immune system to recognize specific cancer antigens, very similar to how traditional vaccines work. Then adoptive cell therapy, which I'll talk about in more detail shortly, involves isolating and engineering a patient's own immune cells to enhance their ability to target cancer. And this personalized approach not only aims to destroy existing cancer cells, but also helps prevent recurrence by providing a long-lasting immunity against that tumor. Immunotherapy has already shown incredible promise in treating cancers that were once thought to be untreatable, particularly in cases like metastatic melanoma or blood cancers such as leukemia. Now, adoptive cell therapy is a transformative approach in cancer treatment designed to harness the power of a patient's immune system to fight cancer. This technique involves collecting a patient's T cell, modifying them to enhance their cancer-fighting ability, and then reinfusing them back into the patient. There are two primary forms of adoptive cell therapy: CAR T cell therapy and tumor-infiltrating lymphocyte, TIL therapy. So in CAR T cell therapy, T cells are genetically engineered to express a chimeric antigen receptor, or CAR. This receptor is specifically designed to recognize cancer cells, enabling T cells to detect and attack them more effectively. And one of the main challenges in cancer therapy is that cancer cells can sometimes disguise themselves, preventing the immune system from identifying and destroying them. So by adding CARs to T cells, we can reprogram them to overcome this disguise, making it easier for the immune cells to target the cancer cells more accurately. And this approach has shown remarkable success, particularly in treating certain blood cancers like leukemia and lymphoma. And clinical trials are ongoing to determine its effectiveness for solid tumors like breast and brain cancers, which present unique challenges due to the tumor microenvironment. So now that we understand the background for cell therapies, let's go a little deeper into the development part of these cell therapies. So in the field of cell therapy, there are several types of cell-based treatments, each with distinct mechanisms and targeted approaches. So on this slide, I am outlining the primary classes of cell therapy, which are widely researched and utilized in cancer immunotherapy. So starting on the left, we have CAR T cell therapy. CAR T cells, which are basically T cells that have been genetically modified to express a chimeric antigen receptor, which is specifically designed to recognize and bind to a target antigen on the cancer cell surface, like I mentioned before. But unlike some other T cell therapies, CAR T cells do not require intact antigen presentation on cancer cells. However, the target antigen must be a surface protein. And CAR T therapy has shown great success, like I mentioned, in treating blood cancers, and there is also ongoing research that is aiming to extend its efficacy to solid tumors. Next is the TCR T-cell therapy, which stands for T-cell receptor engineered T-cell therapy. So here, T cells are modified to express a T-cell receptor, or the TCR, that targets specific antigens presented anywhere within the cancer cell. Unlike CAR T cells, TCR T cells do actually require intact antigen presentation. And this requirement can sometimes be a limitation, as certain tumors develop resistance by altering antigen presentation mechanisms. Nonetheless, TCR-T therapy is being explored as a potential approach for targeting a broader range of cancer types. Now, the third category is the other T-cell therapies, which include methods like tumor-infiltrating lymphocytes, like TILs, which are naturally occurring T cells expanded and activated to fight tumors, or even safety switch modified T cells, which include built-in safety mechanisms to control their activity. Another example in this group is virus-activated T cells and also gamma delta T cells. And these therapies provide a range of strategies to enhance T cell response against cancer, each with its own benefits and limitations. Now, moving on to DC vaccines. It's basically dendritic cell vaccines, and in this approach, dendritic cells are expanded, activated, and exposed to tumor-specific antigens. And these dendritic cells then interact with T cells, presenting the antigens to them and effectively educating the immune system to recognize and attack tumor cells. And DC vaccines are designed to kickstart a patient's own immune response against cancer, and they show promise in combination therapies with other immunotherapies. Now, finally, we have the NK cell therapy, in which the NK cells are activated and modified, and if necessary, then recognize tumor-specific antigens. NK cells belong to the innate immune system, and unlike T cells, they don't require prior sensitization to a specific antigen. So this innate capacity allows them to attack a wide range of cancer cells, making NK cell therapy a valuable option, especially when combined with other immune strategies. So when we look at the various cell therapy classes, we see CAR-T, TCR-T, and other T-cell therapies as key players. And each of these therapies involve genetically modifying T cells to enhance their ability to recognize and destroy cancer cells. In addition, dendritic cell vaccines and NK cell therapies are also being explored as options to boost the immune system to respond in cancer patients. Now, this slide illustrates the CAR T-cell manufacturing platform and its interactions with the tumor microenvironment, which can significantly impact CAR T-cell effectiveness. At the center, we have CAR T-cells engineered to target tumor-associated antigens, and for effective action, CAR T-cells must maintain their cytotoxic functions and proliferation. However, cancer cells and surrounding stromal cells create multiple barriers. And on the left, tumor cells upregulate factors like STAT3, survivin, and IDO, which promote the tumor survival and resistance to immune attack. They also secrete IL-10 and TGF-beta, contributing to immune suppression and tumor evasion. Now, the TME also contains suppressive cell types like Tregs, tumor-associated macrophages, and myeloid-derived suppressor cells, which produce inhibitory molecules like PD-L1 and TGF-beta, leading to CAR T-cell anergy, a loss of activity and proliferation. Tregs further reinforce immune tolerance and limit CAR T cell infiltration. Finally, tumor cells encourage angiogenesis via VEGF and other factors, creating physical barriers to CAR T cell infiltration. So in summary, CAR T cell manufacturing platform not only involves engineering CAR T cells to recognize tumor antigens, but also requires strategies to counteract the multiple immunosuppressive signals in the tumor microenvironment, and enhancing CAR T cells to counter these suppressive factors is key to improving their anti-tumor efficacy. As we've seen in the previous slide for the CAR T cell manufacturing platform, engineering CAR T cells involve complex interactions within the TME. However, the effectiveness of CAR T therapy also depends on the source of the cells. Here are two main approaches, autologous and allogenic. Autologous is where cells come from the patient, and allogenic is where cells are sourced from a healthy donor. And each approach has unique benefits and challenges. So here I wanted to compare these two approaches, highlighting their differences in terms of source, clinical efficacy, safety, turnaround time, cost, and product consistency. So starting with the autologous cell therapy, the source of the cells is the patient themselves. And this approach has demonstrated efficacy with approved therapies like Yescarta and Kymriah for certain blood cancers. Since the cells are the patient's own, there's less risk of immune rejection. However, there is a known risk of cytokine release syndrome, or CRS, which is a very serious inflammatory response. And autologous therapies generally have a slower turnaround time due to the need for individualized cell processing, making them more costly to manufacture. Additionally, product consistency can be more variable as each patient's cells differ. On the other hand, allogenic cell therapy uses cells from a donor pool, allowing for potentially faster production and more standardized products. However, since the cells come from a donor, there is a higher risk of immune rejection and graft versus host disease, where the donated cells attack the patient's own cells. And clinical efficacy for allogenic therapies is still in its early phase trials, with durability concerns due to possible immune rejection. But despite these risks, allogenic therapies are faster and cheaper to manufacture, which could make them a lot more accessible than the autologous therapy. In summary, autologous therapies offer personalized treatment with less risk of rejection, while allogenic therapies provide scalability and consistency. Ongoing research aims to optimize both approaches for broader and more effective use in therapy. Now that we have discussed different types of sources of cells, let's discuss the product development part. So whether the cells come from the patient or a donor, their development into a therapeutic product follows a precise and controlled process. So here we'll look at the 5 key stages in the development of immune cell therapies, which are essential for creating a safe and effective product. So starting with cell isolation, which is the first step involving collection of cells from either the patient for autologous or a healthy donor for allogenic therapies. And the isolation of these cells is crucial as it forms the foundation of the therapeutic product. Now, second step is cell selection. So once isolated, the cells are selected based on specific phenotypes, and this step ensures that only the most suitable cells, such as those with high potential for therapeutic effectiveness, are carried forward in the process. Third step is genetic modification, and in this stage, the selected cells are genetically modified to enhance their ability to target and attack cancer cells. For CAR T or TCR-T therapies, this involves inserting specific receptors into the T cells to enable them to recognize and kill the tumor cells. Now, step 4 is cell expansion. So, after modification, the cells undergo expansion to achieve the necessary cell count for therapeutic dosing. This amplification process is conducted in vitro under controlled conditions to maintain cell health and functionality. Step 5 is cell preservation. So finally, the cells are preserved, often through freezing at very, very low temperatures to maintain their viability until they are ready for infusion into the patient. And this step is essential to ensure that the cells retain their therapeutic properties up to the point of administration. So each stage in this process is meticulously designed to produce a high-quality, effective cell therapy product, ensuring that the cells are optimized for safety and efficacy when administered to patients. And this structured approach supports the development of both autologous and allogeneic therapies and is foundational to advancing cell-based treatments in immunotherapy. As I mentioned in the last slide, cell expansion is critical for therapeutic success, and cytokines are used to support cell expansion and manipulate their functionality and durability. So cytokines play a significant role in enhancing these properties, supporting T cell survival, proliferation, and cytotoxicity. And cytokines are vital signaling molecules that modulate immune responses and can significantly impact the effectiveness of CAR T cells. And on this slide, I wanted to highlight various cytokines and their specific strategies to deliver targeted signals, along with their effects on CAR T cell properties. So let's take IL-2. This is incorporated within the CAR construct or used with an orthogonal IL-2, IL-2R system to enhance the T cell proliferation and effective functions. Then IL-7, through ectopic or constitutively active expression, provides CAR T cells with long-lived potential and durable effector functions, increasing their persistence in the patient. IL-9 promotes stemness and effector function maintenance, while IL-15 uses membrane-tethered expression to prolong T cell survival. IL-8 is engineered to enhance CAR T cell migration into tumors by expressing receptors like CXCR1 and CXCR2. IL-10 is employed to restore exhausted T cells, while IL-12 directly upregulates cytotoxicity and helps modulate the immunosuppressive tumor microenvironment. Then there are other cytokines like IL-18, IL-23, IL-33, and IL-36 gamma that contribute to modifying the tumor microenvironment or promoting specific CAR T cell activities. Each cytokine strategy is designed to address specific challenges within the TME, such as T cell exhaustion or limited tumor infiltration, and basically, by tailoring these cytokine signals, we aim to develop CAR T cells that are more robust, durable, and effective in overcoming the defenses of the cancer cells. Ensuring raw material safety is essential in cell therapy, and in this section, we'll discuss the key regulatory standards that govern GMP-grade materials, ensuring they meet quality and safety requirements crucial for effective and reliable therapies. Raw material safety and quality is critical in cell and gene therapies, especially before these materials reach clinical trials. So materials designated as GMP grade must adhere to strict guidelines to ensure that they are safe for manufacturing processes. And key regulations include the USP Chapter 1043 and USP Chapter 92 standards, which basically cover the ancillary materials, growth factors, and cytokines for cell therapy. Similarly, the European Pharmacopoeia, chapter 5 to 12, which sets guidelines for biological raw materials in cell and gene therapies. And then there is also the Chinese Pharmacopoeia's general chapter and the various ISO standards like ISO 9001 and ISO 13485 that outline the quality management systems for safe material production. Now, compliance with these standards ensure that raw materials meet stringent safety, purity, and functionality criteria, which is essential for therapeutic success. And in the next slide, we will delve into the manufacturing standards for GMP grade materials, specifically focusing on how manufacturers uphold these quality and safety guidelines throughout the production process. GMP, or good manufacturing practice, applies to all aspects of pharmaceutical manufacturing, including the raw materials used in cell and gene therapy. While regulatory bodies like the FDA set overarching standards, manufacturers are responsible for ensuring raw material quality through internal processes. And the manufacturing framework is based on the 5 pillars of GMP. Premises and equipment, that basically is ensuring that facilities and equipment are designed to maintain product quality. Then next is people. Well-trained personnel are essential for consistent, high-quality production. Then next are procedures. So very detailed standard operating procedures or SOPs guide each production step. Fourth is process. So each stage from raw material handling to final product testing is meticulously controlled. And the last is the product. So strict quality checks confirm that all the products meet safety and efficacy standards. So together, these pillars ensure that GMP grade materials maintain high levels of consistency, purity, and safety throughout the production life cycle. And having established the importance of GMP manufacturing practices, for the next slide, I will explore Sino Biological's own quality system, which is designed to align with and exceed these regulatory standards. So at Sino Biological, our quality system is designed to meet and surpass regulatory standards, ensuring that all our products are manufactured with the highest quality. And our quality system is structured into 5 key areas. Laboratory control, where we perform stringent tests, calibrate instruments, validate analytical methods, and manage strains and samples according to standardized protocols. Then equipment and facilities. So our facilities undergo rigorous cleaning and environmental monitoring with equipment maintained for optimal performance. Then third, materials system. This includes supplier audits, warehouse management, and procedures for handling non-conforming products. Fourth is package and labeling system. This ensures that all the products are correctly labeled and packaged to maintain integrity. Fifth is the production system. We control each production batch with critical quality attributes and process parameters, ensuring consistent quality. Our commitment to quality control and compliance provides a reliable foundation for supporting cell and gene therapy advancements. Now, developing CAR T-cell therapies require a multi-stage approach, and each step is crucial for creating an effective and safe treatment. Here, we outline Sino Biological's comprehensive solutions that support each stage of CAR T-cell therapy development. For CAR development, this involves selecting the best CAR candidates through scFv affinity characterization, target protein identification, and assay cell line development. We ensure that Only the most promising CAR constructs move forward. Now for the preparation part, preparing the CAR T cells include nucleic acid removal and packaging the lentiviral for gene transfer. We provide GMP-grade nucleases like supernucleases for DNA clearance, ensuring high quality and safe cell preparations. For the next step, cell expansion, we supply GMP-grade cytokines such as IL-2, IL-7, IL-15, and IL-21 to support robust T-cell growth and expansion. Quality assurance systems are also in place, ensuring every step is compliant with GMP, USP, and Chinese pharmacopoeia standards. For quality control, rigorous testing is essential to confirm CAR T-cell purity, potency, and safety. We offer solutions for cell population profiling, non-target cell detection, and impurity testing to meet these stringent standards. And this end-to-end approach enables us to support CAR T therapy development from candidate selection to quality-controlled final products. In addition to supporting CAR T therapy development, Sino Biological provides high-quality GMP-grade cytokines that play a critical role in enhancing cell culture and expanding therapeutic cells. Let's take a closer look at how these cytokines contribute to advancing cell therapy. Sino Biological offers a range of high-quality GMP-grade cytokines that support various stages of cell therapy development. Each cytokine undergoes rigorous quality control to ensure optimal performance and consistency. Here I have taken selected cytokines that are used for CAR T-cell expansion, and also I'm demonstrating the quality of these products. For example, IL-2 is used in cell proliferation assay to stimulate T-cell expansion, and we're also showing the binding affinity measured via octet red system, providing a consistent foundation for cell therapy development. IL-7, which is essential for immune cell growth, here showing high purity and measured activity to support T-cell health. IL-12, known for inducing interferon gamma secretion, which enhances immune responses, making it invaluable in activating T cells. IL-15 and IL-21 both support T cell proliferation and activation, with IL-15 assisting in prolonging T cell survival and IL-21 aiding in sustained immune responses. Our cytokine catalog includes cell culture growth factors, cell activation factors, differentiation factors, as well as GMP-grade specialized enzymes such as supernucleases for DNA clearance. And these products are designed to support cell therapy processes with consistency and high standards, enabling researchers to develop effective therapies. And this commitment to quality and range of products ensures that Sino Biological can comprehensively support cell therapy programs from development to large-scale production. Sino Biological is dedicated to maintaining high standards of quality and efficiency, which is evident across our facilities, certifications, and quality systems. Our rigorous quality control include immunoassay testing, functional activity evaluations, and analytical method validations to ensure the consistency and safety of our products. We also hold multiple certifications, including ISO 9001, ISO 13485, and ISO 17025, as well as GMP compliance certification from SGS. And we also have DMFs filed with the FDA. These certifications underscore our commitment to quality management and regulatory compliance. We have also been recognized as a top supplier for growth factors and proteins, and we were acknowledged for our contributions to SARS-CoV-2 research in 2021, highlighting our role as a reliable partner in the biotechnology field. Our advanced facilities allow us to handle high throughput projects with the capability to produce batches up to 1,000 liters. Our lab and office space spans 355,000 square feet, including a 54,000 square foot GMP certified clean room and laboratory. We leverage high-tech equipment such as HPLC, BIACore, and Octet systems for precise testing and product quality analysis. And our team includes professional quality management personnel and over 900 research staff members, reflecting our investment in skilled expertise and continual growth. With these resources and quality systems, Sino Biological is well equipped to meet the evolving demands of biotechnology, supporting process projects from research to large-scale production. So in developing cell therapies, navigating supply chain challenges and carefully evaluating suppliers are crucial for ensuring consistent quality and reliability. In the last section of this webinar, we'll explore some key challenges and what to consider when selecting suppliers to ensure quality and consistency. Developing cell therapies is a multi-step process that comes with distinct challenges at each stage. But today, I only want to focus on some of these critical challenges that I've highlighted by the red arrows. So one of the primary challenges in cell modification is designing functions that enhance therapeutic effectiveness while minimizing unwanted immune responses. And this step often involves sophisticated techniques such as transgene insertion or genome editing, which must be precise to avoid triggering adverse reactions in the patient. And achieving this balance is essential for safe and effective cell therapies. Then next is scaling up cell production from preclinical to clinical stages is another major hurdle. And consistency between batches, known as batch-to-batch reproducibility, is crucial to ensure each therapeutic dose has the same safety and efficacy profile. Additionally, sourcing of reliable GMP-grade components is necessary to maintain product quality and regulatory compliance. Then the last one is comprehensive testing for potency, sterility, viability, and purity is essential to confirm that each batch meets quality standards before it reaches the patients. And this involves specialized assays such as qPCR and cell phenotyping to ensure that the cells are both safe and effective for therapeutic use. Addressing these challenges is fundamental to developing cell therapies that are both safe for patients and scalable for broader clinical applications. So, as we've seen, overcoming the challenges in cell therapy development requires careful planning and quality control at each step. One of the most crucial aspects of ensuring consistency and reliability in production is selecting the right GMP supplier. Let's now discuss some key considerations when evaluating suppliers for your cell therapy raw materials. So, when selecting GMP suppliers for cell therapy, asking the right questions is essential to ensure that they meet your quality and consistency standards. So here are some questions to ask your GMP supplier. Like, what lot sizes are available and can you purchase an entire lot for consistency? Are there stability records across different lots, and are regulatory documents accessible for each lot? And can a master supply agreement be established to formalize the relationship? And here are some additional considerations that you can apply. Like, should you have a backup supplier to avoid disruptions? It's wise to inquire about GMP requirements as early as possible to avoid any late-stage changes. And if you're scaling up, consider incorporating GMP requirements early in the chemistry, manufacturing, and controlled process to streamline the production. And by evaluating these factors, you can establish a solid supplier partnership that supports the quality and scalability in cell therapy manufacturing. So as we consider supplier evaluation, it's important to understand Sino Biological's own global footprint. With facilities across key regions, Sino Biological offers localized support while maintaining global standards. Sino Biological has established a strong global presence to meet the needs of clients worldwide, with subsidiaries and R&D centers across North America, Europe, and Asia. We ensure localized support and quick response times. Our major facilities include our Center for Bioprocessing in Houston and SignalChem Biotech facility in Vancouver, Canada, that we acquired in 2024. And this network allows us to provide direct sales and support in over 90 countries with a team that understands the local and global biotech landscapes. With this global infrastructure, Sino Biological serves as a leading supplier of reagents and services. Now let's look at the breadth of products and services available to support research and development. So Sino Biological is a leading supplier of reagents and offers comprehensive CRO services. Our extensive catalog includes over 9,000 proteins, over 14,000 antibodies, over 49,000 genes, and over 600 ELISA kits. And we also offer services such as recombinant protein expression, recombinant antibody production, as well as antibody development for both monoclonal and polyclonal antibodies. With over 10,000 citations, including publications in journals like Nature and Science, Sino Biological's products are widely recognized and used in top-tier research worldwide. And Sino Biological's reputation in the field is reinforced by industry recognitions and awards. These honors highlight our commitment to quality and innovation in biotech. Sino Biological's excellence is reflected in numerous industry awards, and while COVID put us on the map with numerous awards related to viral research field, here I wanted to highlight only the relevant awards to this webinar. So recognized by CiteAB as a protein supplier to watch in 2022 and a growth factor supplier to watch in 2024. We have also been listed among the top 100 healthcare technology companies of 2022. Also awarded for performance by Scientist.com and acknowledged for contributions to cancer immunotherapy solutions. And these honors highlight Sino Biological's commitment to quality, innovation, and leadership in the biotechnology sector. Sino Biological is dedicated to supporting cutting-edge cell therapy through global resources, high-quality products, and reliable expertise. Please feel free to reach out to our team at any of our U.S. locations for further inquiries or collaboration opportunities. We look forward to advancing innovation together. And that concludes my presentation. Thank you all for your time and attention today. I would be happy to take any questions that you may have.
So thank you for that wonderful presentation. We will now open the floor for some questions. So please remember to send us your questions using the chat feature. So, first question, so what challenges have you encountered in developing GMP cytokines and how have you overcome them?
So developing GMP-grade cytokines involves significant challenges. Off the top of my head, I can think of a couple of challenges that I can talk about today. So the first one, I think it may sound a little strange to somebody who has not been involved in cytokine production, that some of the properties of cytokines are similar to endotoxins, making it difficult to reduce the level of endotoxins during production. So basically what we have done is we try to find the differences between this type of cytokine and endotoxins by screening different types of purified fillers so as to achieve the purpose of removing these endotoxins. And some cytokines are actually not very stable. So after a period of time, the proteins will aggregate and their activity will decrease. So we basically screen and optimized these formulation of this type of protein through experiments to select a formulation suitable for it, basically to achieve the purpose of stable preservation of this product. And another one that I can think of is that some of the cytokine expression levels are relatively low, which leads to the need to scale up to a large volume for industrial production. And basically, to address this issue, we generally optimize the fermentation process by improving cell density through feeding and perfusion, thereby increasing the expression level of the product. So, all in all, developing GMP-grade cytokines require a lot of patience and a lot of troubleshooting to optimize the production pipeline to reliably produce big batches of these cytokines.
Perfect. Thank you very much for that answer. We have another question. So, how do you see the future of cell therapy and how will Sino Biological play in driving this innovation?
Oh, I love this question. So the future of cell therapy is incredibly promising. With ongoing advancements in CAR T, TCR, and NK cell therapies, among like a lot others. So these therapies are moving towards more personalized and scalable solutions with an emphasis on reducing costs and improving accessibility. And Sino Biological is well positioned to drive this innovation by supplying high-quality GMP-grade cytokines and offering CRO services for reagent development and testing. Our extensive catalog of research-grade and GMP-grade products, as well as antibodies for T-cell activation, for example, these support both early-stage research as well as large-scale clinical applications, basically enabling us to contribute to the entire pipeline of the cell therapy development.
Fantastic. Thank you. Another question. So, what advanced equipment and quality control processes does Sino Biological use to ensure the consistency and safety of its products?
So Sino Biological basically uses advanced equipment like HPLC, malts, BioCore, and Octet systems to ensure precise quality or quality control in the product development. And these systems help us to evaluate purity, binding affinities, and other key characteristics of cytokines and other reagents. Our facilities adhere to GMP and ISO standards with comprehensive quality management practices, including batch records, critical quality attributes, and microbial control. Our high-throughput testing platforms for immunoassays and functional evaluations further ensure that each product batch meets stringent safety and efficacy requirements.
So, how does batch-to-batch reproducibility impact the clinical efficacy of cell therapies? And what role do GMP grade components play in maintaining this consistency?
So batch-to-batch reproducibility is essential in cell therapy because even minor variations can impact the efficacy and safety of the treatment. Also, as I mentioned in the answer to the previous question, that when you're running a large-scale clinical trial, it's very important to evaluate your GMP-grade product supplier to basically on batch-to-batch consistency and the capability to produce big batches. Now, GMP-grade components like those provided by Sino Biological are produced under strict controls to ensure each batch meets the same standards for purity, potency, and safety, and by maintaining high standards for reproducibility. GMP-grade components can basically help ensure reliable therapeutic outcomes across multiple batches and patient treatments.
Thank you very much. Another question. So, in your experience, what are the most common questions that people ask you when they are evaluating suppliers?
So, the common questions include questions about batch size capabilities, of course, and then also availability of the regulatory documentation and whether the clients can audit the suppliers or not, and at what frequency even. Then also stability data availability and also quality certifications such as GMP compliance that we have with SGS. Our clients also frequently ask about the scalability of production and also the lead times of the production as well. Another key area of interest is the testing and validation processes we use to confirm the product quality and consistency. So those are the main questions that I can think of that we have received in general.
Brilliant, thank you. So somebody else has asked, so how long does it take to conduct a thorough supplier evaluation for cell therapy materials? And what key questions should be asked during this evaluation?
So that really depends from client to client, but a thorough supplier evaluation will typically take like several months to sometimes even a couple of years. And it depends on the complexity of the materials and the regulatory requirements in general. So the key questions that you should be asking your GMP grade supplier that you're evaluating at the moment should be, of course, like what quality certifications does the supplier hold, like ISO, GMP, etc. What documentations can they provide on batch consistency and stability? Then are there validated testing methods for purity, potency, and safety? Then also what are the lead times and logistics for large scale orders? That's very, very important. Can the supplier provide a master supply agreement and secure regulatory documentation for each batch? Conducting a thorough supplier evaluation ensures that the materials meet the necessary standards and that supply chains are reliable and robust.
Perfect. Thank you very much for answering those questions. So I believe that this draws the webinar to a close. Thank you so much for your questions. And once again to Ritwika, as mentioned, the on-demand recording will be made available over the coming days. So please look out for this in your inbox. Thank you, everybody, and have a lovely day.